were studying chronic myeloid leukemia, which, although is a disease that has been basically the poster child of molecular targeting therapies, because it was the first one really to show how tyrosine kinase directly to the BCR-ABL, that is the fusion gene that is the hallmark of this disease, can, this aberrant tyrosine kinase can be targeted by a tyrosine kinase inhibitors with in this patient that in two decades ago were treated with allogeneic stem cell transplantation, now switch completely in terms of the treatment, because they basically just with the pill a day, they'll be able to achieve remission and long-term survival. However, many of these patients, if they stop the treatment, eventually may relapse, or if they're intolerant to the treatment, they cannot take it, they eventually relapse. And some of these patients even transform in something called blast crisis, the equivalent acute leukemia. So we wanted, and the reason why they do that, because there is leukemia stem cells, these evil mother, so to say, of all the leukemia cells that persist despite the treatment. So we want to understand what was the mechanism by which eventually the patient, the CML transform into blast crisis, the chronic phase transforming blast crisis. And looking for a different mechanism, we've been long time interested in microRNAs. And basically we found that patients with the blast crisis, with respect to patients with the chronic phase CML, they have a lower level of miR-142. And miR-142 has been, as a track record in the literature, as a potential regulator of the molecular switch. There have been other groups that have reported that in innate immunity, dendritic cells cannot be switched and activated one if they are deficient of miR-142. And this is because the oxidative phosphorylation cannot be switched in the glycolysis, a different metabolic pathway to produce energy. So because this finding, we cross the mouse with the chronic phase CML with the mouse that have a deficit of a miR-142. And our hypothesis is that these mice could have eventually transformed. These mice that usually do not transform from chronic phase in blast crisis, by missing miR-142 could have been transformed in blast crisis. And that is exactly what happened. When we cross these mice, we obtain a progeny with both the BCR-ABL and miR-142 knockout. These mice develop a blast crisis CML. So, and the most important things, when we did a secondary transplantation, took the bone marrow from the initial BCR-ABL and miR-142 knockout mice, we took those bone marrow, we transplant in normal mice. The normal mice develop blast crisis too. So this means that miR-142 had transformed chronic leukemia stem cells in blast crisis leukemia stem cells. The third finding that we have, we thought, okay, now if these mice miss miR-142, then what about if we make a synthetic form of 142 and we give to these mice? Are we able to return this mice to the initial phenotype that is a chronic phase, or even if we combine that with tyrosine kinase inhibitor to cure these mice? And that is exactly what happened. When we treat the mice with the synthetic miR-142, these mice did not produce blast crisis, or if they were in blast crisis, they returned to the chronic phase. And when we were able to, combined the synthetic miR-142 with the tyrosine kinase inhibitor, that is the standard of treatment for BCR-ABL leukemias, we found basically that these mice survive much longer. Some of these mice were, in fact, even cured. And when we did a secondary transplantation, we realized that, we were able to completely erase leukemia stem cells. Basically, just by changing the metabolism, we were able, from glycolysis to oxidative phosphorylation by deficit of miR-142, we were able to transform the phenotype of the leukemia stem cell from a chronic phase leukemia stem cells into blast crisis leukemia stem cell. No, I will say not much challenge because we are, we have been studying microRNAs for a long time. The challenges that have been for the community has been a little bit skepticism about the use of microRNA as a therapeutic, in general RNA therapeutics. But we see that in the last few years, the RNA technology and RNA therapeutics as a class of new drugs is gaining very rapidly. I would say gain the confidence of the community. In fact, if I'm not mistaken, for instance, the Nobel Prize for medicine this year was given to 2 people that have used RNA-based techniques to produce vaccine. We have treated probably around more than 200, 250 mice with this drug. We've done also some toxicity, preliminary toxicity study. We have not seen any toxicity so far. We hope that we have a new drug that will help to, in combination with tyrosine kinase inhibitors, will help to, one, eventually prevent the transformation, but also treat the transformation of this patient blast crisis. 2, we hope that perhaps with additional studies, the combination of the miR-142 synthetic that we call mimics with the TKI are able to completely eliminate leukemia stem cells and allow these patients that usually, even for those that do well, they are on lifetime treatment with tyrosine kinase inhibitor, eventually to discontinue it for the lifetime, their treatment. And most importantly, this could be also used as a paradigm to understand what is the Achilles heel of leukemia stem cells. And again, we have seen with the synthetic miR-142, we're able to fragment and destroy the mitochondria of leukemia stem cells. And we have a lot of hope that we can use this drug also in AML, other type of leukemia, perhaps other type of cancer, for which there is cancer stem cells based biology, so to say.